time to talk. So welcome everyone. I'm Julia Bailey-Saris, and I'm a professor in botany and plant sciences and the director of UCR's Center for Plant Cell Biology. I'm also, fortunately, also the director of our NSF-funded Plants 3D program, and I know a number of the members of the audience are the trainees of that program. One of the initiatives of CEPSUB is to bridge between plant biologists, plant microbial biologists, and others on our campus. We have a history of working together with chemists, computational scientists, and more recently with engineers. And the Plants 3 program really is this opportunity for students to receive additional training in a discipline outside of their major one, be it plant sciences or even environmental sciences or chemical engineering, such that we can all participate more actively and effectively in finding solutions to the needs of humans in terms of crop production, food preservation, and also health needs. And so it's interesting that on our campus we have a second center, and that's the CAFÉ Center that's run by Norm Elstrand, who's sitting here in the room with his assistant director, Quinn McFedrick, and also Deborah Pagliacci, who is the real director of the program. CAFÉ, let me tell you, is similar to CEPSUB in that it is a multi-college initiative, and their focus also is to bridge across disciplines in this arena of food and agriculture. But they bridge a bit further than us, considering social sciences and public policy. It's a very open group of individuals where graduate students, postdocs, faculty are all encouraged to become members by going to the CAFÉ website and signing on. And this will allow you to have notifications about their activities, which include things like hosting seminars, such as this one today, as well as other fora, workshops, and even, when it is possible, actual conferences. CAFÉ is also part of UC's Global Food Initiative, and so really links UCR with others across the University of California who have this topic front and foremost in their minds. So today, because of CAFÉ's idea of having really distinguished speakers placed in different departments this year in their seminar series, we have the honor to co-host Benedetto Marelli, who is an associate professor from MIT's Department of Civil and Environmental Engineering. I imagine that most of you have not heard of Professor Marelli's work, but I will tell you we are in for an absolute treat. He was trained in biomedical engineering at the Polytechnic University of Milano before he joined McGill University, where he did a PhD in materials engineering. From there, he went to Tufts University and joined a group which is called the Silk Lab. And the Silk Lab does a lot of things with silk as a polymer. And what we're going to hear is how Marelli's work combines material sciences of polymers such as silk and seeks applications particularly in the agriculture and food space. I believe he's going to tell us about what is really designs of different polymers, the testing of those, and then their applications. And I just want to reiterate, for those who are not familiar with why we call our graduate training program Plants 3D, it's because of this discover, design, deploy theme that really is at the heart of Benedetto's research. So I think we can just, I will just pass the baton first to Deb, who has a comment to make, and then to Benedetto so that he can tell us about the type of work that he is doing that potentially can help protect food from post-harvest demise due to pathogens, or even have applications in terms of citrus greening disease, which is obviously something that's a topic of research for a number of you here. So first Deb, and then Benedetto.
Thanks, Julia. I think you really said everything about CAFÉ, so I'm not going to have anything, but I wanted to make sure that people know that there is a Padlet, which is a layout wall to basically put any comment you want about this presentation. CAFÉ work on bridging discipline, bringing people together. So we would like to know what do you think about the innovation? What do you think is the greatest barrier to implementation? Or as a scientist working in a different discipline, or even as a community member or as a student, how could you engage in this research to improve positive impact? So you can open the Padlet on the side and you can add any comments you want. And thank you very much to Benedetto for accepting our invitation. And we can start.
Just one last comment. I welcome you to add questions in the chat box during the seminar. And at the end, we'll allow graduate students and postdocs to ask the first questions.
So thank you very much, everybody, for the nice introduction. I will dive into the talk, so I'm sure I'm covering everything in the amount of time I have. Today, we're going to learn about silk, and we're going to learn about how we can use this material, which is very ancient, to innovate in food and agriculture and hopefully having a positive impact on our society. My background, we already covered it. My lab does three things. We do synthetic biology of structural biopolymers. Then we direct their assembly, we do nanomanufacturing, and eventually we use these new materials to find innovation in food and agriculture. And today we're going to focus on the second and third topics. The mission of my lab is to find small solutions that can have a big impact when it comes to sustainably feed 9.7 billion people. And the reason why we're doing it is because, as you heard before, also during the introduction, we're going to add 2 billion people. We already have that 800 million people are suffering from hunger. Many of them, unfortunately, are also in the United States. The food and agriculture system emits a lot of anthropogenic gas. And particularly something that my lab is really passionate about is food waste. So we're wasting a lot of food. Depends on the geography in different parts of the supply chain. But the food that is lost and wasted is really a big polluter of the world. So it has a very, very high impact on our society, to the point that it's the third producer of greenhouse gas emission after China and the United States. And at the same time, 25% of the fresh water is lost with the food that we're wasting. So how do we innovate in this sector? We start from the entity that you see in the middle of your screen, which is a silk cocoon. Okay, so a cocoon is the starting point. So it's a raw material, is the byproduct of agriculture, if you like, is the leaf. Is a leaf that has been converted by the silk caterpillar into a protein. And eventually we reverse engineer the cocoon. So we take this fiber and we go back to the same state that it has in the gland of the caterpillar or of spider, if you want to use spider silk. So we have a water suspension. So the moment we're able to use chiotropic ions to disgregate the hydrogen bonds that keep the fiber together, and then we leach out these chiotropic ions, which is typically lithium bromide, for example, we end up having a water suspension of silk. And once we have this water suspension, then we can engineer new material format. So forget about silk as a fiber. You experience silk as a fiber because the caterpillar or the spider needs it in a fiber format for their function. But in reality, silk is a bioplastic. We could consider it like a bioplastic that comes together, and we can have several material format. These are all the materials format that I contributed engineering throughout my career. So we can make screws out of silk by having blocks that you can machine down. We can make microneedle, particle, films, gels, tubes, and we can advanced manufacturing. We can use, advanced nanomanufacturing technique to make advanced materials that can compete with the synthetic material with the big benefit that we're using a protein. So at the end of the day, it's a biodegradable material that comes from a leaf and also can be easily interfaced with living systems. So how do we work with silk? What happens? How can we engineer these advanced materials? Well, we start from the water suspensions that you see here, and if you take an electron microscopy, what you get are these nanoparticles of silk that eventually, in a gel transition coalesce to become a nanofibrillar matrix. And then in the gel to solid transition, the empty spaces in the gel, which is water, is replaced with inter and intramolecular hydrogen bonding, so that the material really comes together to make a monolithic material. And the beauty of silk is not only in the mechanical properties that you may already know about, but is in the fact that the protein can be engineered in two different states. One that is amorphous, is called amorphous silk, which is a random coil silk, so which is completely water soluble, or it can be ordered in beta sheets. So these are insoluble, water insoluble proteins. So the same protein is called polymorphism, can be water soluble or water insoluble, and this allows us to have a very wide degree and freedom for fabrication perspective. So the process that I just told you is basically a disordered to ordered transition of silk, but for some reason I never really liked it because it's very difficult to guide the assembly without using top-down fabrication techniques. And so my student, PhD student Hui-san, she came up with a new way to nanofabricate silk. And we started from nano whiskers. So we just concentrate on this TM. So nano whiskers, the scale bar here is like 100 nanometer, are these nanoparticles. And if we mix these nanoparticles with the silk solution, as we have here, we have that over time, the silk start to assemble around the whiskers to make nanofibrillar structures. So as you can see, a disordered material starts to become an ordered material where we can regulate it very tightly, the folding and the assembly on silk. The process can be modeled with the Avram equation. I'm not going to spend too much time on this. The Avram equation is the typical equation that is used in phase transformation. And as a result, we can eventually get materials with different properties. So we can control the mechanical properties. As we see from this AFM analysis, we can control the molecular folding. So whether we get alpha helical structure, beta sheet, or random coil structures. And this allows us to have a lot of freedom for fabrication, but also to topographically control how the silk assemble, both at the morphological state, so basically if we get nanofibers compared to other structure, but also the molecular state. So are we getting a protein that is mostly beta sheet or a protein that is mostly alpha helical and beta sheet? And the difference between these, the different folding are differences in the physicochemical properties of the protein, typically on the membrane properties. So how well we can control gas permeation, water permeation, how it reacts to bacterial contamination and such. And we can then think about using these nanofibers in combination with top-down fabrication techniques to 3D print or to 2D print our materials such that we can have what are called hierarchically ordered structures. So materials that are organized in an isotropic way from the molecular scale to the centimeter scale. So this is just quickly to convince you that we know and we're learning every day more how we can fabricate silk. But then once we know how to grow silk materials, what are we going to do with it? Where I'm going to go relatively quickly through these 4 applications that we developed that might be of interest for the community. So the first one is we engineer seed microenvironment. So there is a big need to somehow go beyond the fertilizers and how we envision fertilizers nowadays. Fertilizers are fantastic. Synthetic fertilizers are fantastic. Ammonia-based, so nitrogen-based fertilizers allow us to exit famine in many parts of the world. The Haber-Bosch reaction that is used is, however, very, very energy intensive to the point that almost 3% of the CO2 produced in the world and the energy consumed goes in the Haber-Bosch reaction to make ammonia. And so an alternative to this is to use biofertilizers, which are basically microorganisms that live into the soil and that can be beneficial for plants. And so this is why they're called plant growth promoting microorganism, or in the case of bacteria, plant growth promoting bacteria. There's a lot of research in this area, so we very humbly approach this research area from a material perspective. And once we were doing, so when we were doing the literature review, and particularly my PhD student Augustine is the leader of this project, we noticed that there was a big gap between people who are using bacteria and people who are using plants, and the gap is how we deliver the bacteria to plants. And so this is how we want, and to the soil in particular. And so this is basically the problem that we try to tackle. How can we successfully deliver these biofertilizers into the soil, particularly because they don't survive well desiccation, they don't survive well outside the soil. And so typically the stream process is that we isolate bacteria from the soil, we purify, we grow them, and of course we're isolating the ones that are beneficial for plants. Then we make a formulation of the biofertilizers, which includes the bacteria and some material. And then eventually we applied it either on the surface of the seed through priming or seed coating. We can apply to some extent as a foliar spray or as a soil injection. However, from a material perspective, the most important and interesting component for delivery would be the seed coating, particularly because currently in the seed coating, we mostly have color and protectant, and we do have a very small amount of biofertilizers. So we saw an opportunity here in how we could design new materials that can change the microenvironment of the seeds such that we have a successful delivery of these biofertilizers into the soil. So we started the project 5 years ago. We published the first paper 2 years ago. And particularly what we developed is a biopolymer that is made by silk and trehalose. Trehalose is a disaccharide that is present in nature in many organisms that resist desiccation or harsh condition. You can think about tardigrades, you can think about some plants that produce trehalose to relieve stress. And so we made this mix of silk and trehalose, and we designed a seed coating. We looked at rhizobium as our model. So basically a bacterium that can infect legumes and can fix nitrogen for them. And then we used the common bean as our seed model. We started by looking at how well we could preserve this rhizobium, particularly we use Rhizobium tropici. And we did see that our formulation with silk and trehalose was able to outperform over time the current standard used in the industry, which are as polymers PVP and methylcellulose. And these are the materials typically used for coating or to deliver biofertilizers. As you can see, we do have almost a log of loss after 1 week and almost 2 logs after 3 weeks. So we do have a big reduction in the viability, but this is still enough to, we're going to see that it's still enough to guarantee the successful delivery of the biofertilizers into the soil. And these are colony counting results. We don't have the opportunity to test this in Massachusetts. So if anybody would be interested in helping us testing these coatings in the United States, we would really appreciate collaborating. The collaborators that we have are in Morocco. So we shipped our seeds with the FedEx, basically plane and then truck to Morocco, to Ben Guerir specifically, which is between Casablanca and Marrakesh. And we started doing experiments with Moroccan soil that was affected by salinity. So this was a fertile soil that eventually became too saline to be productive. And we did see a big difference in the germination and in the growth of the bean seeds by applying our coating. So after this publication, we then decided to move forward. And as our collaboration with Morocco was growing, they were asking us to make coatings that help alleviate drought stress and water stress. And so on top of the silk trehalose coating, we added a new coating, which here is called layer 2, where we, and to do so, we were inspired by basil and chia seed, who produce mucilage. And so we basically designed our own mucilage that would basically make the second layer of our coating. And here again, the second coating is not only thought to absorb, as you can imagine, water from the soil, but specifically to be sure that the biofertilizers are well preserved and they can establish a strong colony in the soil before they are exposed to the outside world, which is very harsh, if you like, of the soil. And so they have enough time to establish a successful colony that can eventually infect the plants. So to make a very long story short, we defined the best coating by changing several parameters. We investigated the mechanical properties, the rheological properties of the coating to be sure that it satisfies the requirement of the seed coating industry. We did some preliminary tests to see how the bacteria, in this case, the Rhizobium tropici, is producing GFP, so we can visualize in it how it performs once it's rehydrated. So in this case, this is a 2D study, if you like, where we're using a transmembrane. And the first layer is silk with tropici and trialose. And then this is our second layer that would absorb the PBS. And then the water goes in the first layer dissolves, the bacteria are released. And by time they make strong colonies in the gel. The gel also acts as a source of nutrients, as a carbon source, so that the bacteria can make a strong colony and eventually can start to infect the plant. And our preliminary experiments in my lab showed us that we did, in fact, make nodulation. So we did, basically the coating was successful in inducing nodule formation. And eventually when we moved to Morocco and we did experiment in an experimental farm, still in pots though, we were able to test different type of water stress for the soil. And interestingly, we had, with the 2-layer coating, we have a higher yield, a higher biomass, a longer root system. And particularly the 2 data I'm presenting here today are the total phenolic compounds, which decreases, which means that basically the plant is less stressed. And also the stomatal conductance increase, which means that the plant has, again, is another indicator of being less stressed by drought condition. And of course, I'm not a plant scientist, so if you have any question regarding this, I'll try to answer as well as I can, but I have my limitations when it comes to complete understanding this data. So we then moved to another part of Morocco that is called Ayoun, which is where it has very high salinity and very little water. And now we're conducting field experiments. And unfortunately, this experiment needs to be postponed 1 year, so I cannot present you all the data, just a flavor where you can see that we do have a big difference in the yield, typically, so in the formation of fruits, between the plants that are non-coated and non-treated and the one that are coated with our technology. And this technology, Augustin, so the student was graduating in 2 weeks. He's really willing to bring it to the next level. So he's going to start a company out of it that is called Evo Bio. And that is going to be focused on the delivery of microbes in the soil. And so if he's hiring, so if you want to join, he's welcoming basically application for this just established startup. The second project I would like to talk to you about is Phytoinjectors. So we're going from basically boosting seed production now to a fully formed plant. And so what we wanted to do is still, once again, resolving a delivery problem. But the delivery problem we want to solve is how do we deliver any type of active molecules in plants without using foliar spray, because foliar spray is amazing, growers loves it, but it has strong limitations. The typical limitation are that you have tissues that you cannot reach, and citrus greening, which was previously mentioned, is basically one of these. It's one of these situations where, for example, with foliar spray you cannot reach the phloem, and so it's if you have a bacterium that is restricted to the phloem of the xylem, are very difficult to treat. Additionally, you have a lot of runoff when you apply fertilizers of pesticides, typically pesticides, on the leaf. You have almost a 98% runoff with big environmental and health conditions that you need to mitigate. And so we were looking at new ways to deliver materials in plants, and we came up with the idea of using microneedles. So what you see here is a microneedle made out of silk. The silk here has been engineered to work specifically with plants. So we had to increase the amount of hydrophilic amino acids in silk such that it could degrade in plant. And in this case, we can load the microneedle with payload, and we can reach different plant tissues to make a precise delivery in plants. Jan Tenkau, who's my PhD student, he developed different microneedles with different shape, geometries, surface roughness to address different plant tissues. And particularly, we looked at the apical meristem, we looked at the phloem, we looked at the xylem, and we looked at the foliar tissues. As I said, in order to be sure that we can reach the vasculature, we looked, in this case, for example, we used the tomato as a model. And so we looked at the histological section of the tomato. We took measurements, how deep we should go, what would be the dimension of the tip in order to be sure we're targeting the xylem or the phloem. And so we designed microneedle around these anatomical measurements. And also we wanted to be sure that the microneedle were strong enough to pierce through the plant, because the plant is very compliant. So the plant tissue is very compliant. So you need to be sure you're strong and sharp enough to penetrate and to go into the right, to penetrate through the epidermis and to go into the right, the cuticle and the epidermis, and to go into the right tissue without compromising the performance of the microneedle. As you can imagine, this technology works very well when we have a very geometrically organized distribution of our vasculature. If you go towards monocots, where the vasculature is mostly randomly organized, we're becoming less efficient. I also want to mention as a drawback, we do have a little bit of killing zone around the injection site. Now we're reducing this killing zone with the new technologies we're developing. And we do have a little bit of dryness post-injection. However, if we leave the back of the microneedle behind, or if the microneedle is not completely water soluble, this dryness that you have is strongly reduced. We don't have the formation of callus using silk. If you're making microneedles with other materials that are non-compatible with plants, the plant reacts typically and make the callus. As I said, we're designing new silk, so we had to study and investigate the drug release profile to be sure we had a sustained release in plants. We used different payloads, and we investigated the release using simulated SAP, and we find different profiles. This is just a good guidance for us to know how to engineer silk when it comes to deliver payloads into different plant tissues. And finally, we went into plants. And so the first experiment I want to describe with you is where we deliver fluorescent molecule to the vasculature of a tomato stem. I think this is a 4 weeks, if I will remember, tomato stem. So we target the xylem here. This was the injection site. And then we went from source to sink, which is basically from canopy to roots in this case. And we did see that the fluorescence was going down towards the roots. And then we did the vice versa. So we injected the xylem, and in the xylem, we did see that that our fluorescence was going mostly towards the canopy. However, due to random work and due to the fact that you're slightly modifying the hydrodynamics of plants at the beginning, you do have some basically a random walk of your fluorescence towards the root as well. And then finally, we were able to model this data. We developed a very simple 1D model. I'm not going to go through it, but we used the model, particularly the diffusion constant and the velocity, to instruct the next studies, which were made with luciferin and luciferase. So we used the luciferin luciferase model, which you might be familiar with, is a bioluminescent model, to test whether we could have the bioluminescent reaction happening in the plant tissue by delivering, for example, here, luciferin and luciferase. And so we delivered those, and then eventually the protein and the molecule and the enzyme, they mixed into the tissue of the leaf, so in the leaf blade, and the leaf was lighting up in the dark. We also modified our microneedle not only to deliver, so not only to degrade and deliver the payload, but also to absorb the plant sap, so as a sampler. And so to do that, for example, we injected luciferin here, and then we use a luciferase-loaded microneedle that was absorbing sap. And as you can see, the tip of the microneedle was lighting up after removing the microneedle, because it was basically absorbing the luciferase that we delivered previously in the plant. We did test the dynamics of the reswelling just to be sure we were doing things correctly from a material science perspective. And so we finished that application. And then the last application for these phytoinjectors that we tested was about plant engineering. And we did it in the most simple way. So we deliver agrobacterium to a tobacco model, particularly the tobacco. And so this is a plant that was developed by Jinko Wang at the Whitehead Institute. So it's a plant that basically doesn't express GFP, but then once it's infected with the agrobacterium, starts to express GFP. And so we injected the apical meristem, and as you can see, we have GFP production in then once the leaf was formed. Or we also injected at later stage, and what happened at the later stage is that the transformation of the plant only happened locally at the injection site. So I think what was exciting for this study, at least for us, is that we were able to inject the apical meristem, or the first foliar, if you like. We did see a bit of a scar that we left here. So this is why maybe this specific image refers to the first foliar and not to the apical meristem, but we were able to produce GFP throughout the plant. So sorry, throughout the new formed leaf. Many people then asked me whether this could be used in agricultural settings. At the moment, we're mostly exploring, together with Professor Michael Strano, the use of these plants in the urban farming settings. But in the case of agricultural settings, we can envision that these microneedles can be mounted on autonomous vehicle that eventually could apply the needle into plant on a field. And just to give you a flavor of what we try to develop, Yanteng did an experiment that I think only worked once, but he mounted like a toy gun. He used a toy gun, he mounted it on a drone, and then he loaded the microneedle as a bullet, if you like, and then using a computer vision system, he shoot the microneedle, and he was able, I think only once really, to hit the target. Now, applying this in a real field with wind and everything would be very difficult, particularly with the current technology, but at least gives an idea that we can think about autonomously deliver this microneedle into plants. The same microneedle can also be used for food. And so now we're gonna leave the plant and we're gonna move towards the food. And particularly for food, we can use this microneedle for food sensing to detect pathogens. Pathogens and foodborne pathogens are a very big problem in our society. At the moment, for example, there is an unknown outbreak of salmonella. It costs a lot of money to the US economy. Many people get sick. Unfortunately, some of us also die from foodborne illness. The technologies to sense food are largely available. Most of them are disruptive, or most of them can just sense the surface of the food, and they're missing sometimes the pathogens that are inside the food. And so in order to avoid this, we basically use the same microneedle that I described you before as an extractor of fluids from food, such that the fluids can then be presented on the back of the microneedle, where we print an ink, so a sensor that can sense and report the presence of specific pathogens like salmonella or pathogenic E. coli. And to do that, we develop a bioink that changes in color from blue to red when the antibody detects the bacterium or its toxins. The major plus of this technology, if you like, is that silk is now considered generally recognized as safe material, so has the GRAS status due to the work that my lab did, and we're going to discuss this later on. And so in theory, silk is a food contact material, and you can think about if you have your food packaging, the microneedle can go through the packaging in contact with the food, and the bioink is on the outside. So that bioink can be a non-food packaging material, and your silk microneedle can become an interface between the food and the sensor through the packaging. So we developed the bioink. So we looked at basically different concentration of antibody, the time that would require for the color change. We engineered the microneedle to make them microporous such that they could really absorb water pretty fast. Sorry, water, the fluids from food very fast. And eventually we tested it. So in this case, this is a hake fillet that was spoiled with pathogenic E. coli. And and then was packaged and sealed under vacuum. And then we use our microneedle to pierce through the packaging. And as you can see, after a while, in this case, this is a long time, 16 hours. Now we're faster. We had the color change between blue and red. So we recognize the presence of the pathogen. And so once we develop this technology, now the next step would be to embed in each microneedle a different, basically, sensor. So we're going to try to make microneedles that can sense different pathogens all at the same time. So basically, for example, here, each letter would sense for a different pathogen. And then the last part of the talk, and I'm going to try to be fast so we have time for the Q&A, is about food coating. So what started it all? So how my background is in biomedical engineer. Before I worked on implantable optoelectronics. So how did I got interest in food and agriculture is through the experiment that you see in the middle of the screen. We were having a lab competition where we had, when I was a postdoc, where we had to cook with silk because we knew that silk was edible. And so I tried to do the equivalent of a chocolate fondue strawberry, but using silk instead of the chocolate fondue. And nothing happened. Like, I dipped my strawberry in silk, I left it on the bench, and nothing happened. And then I left. I came back 1 week later at the same bench, and I actually figured out that the half of the strawberry were spoiled and the other half were non-spoiled. And then I realized that basically the silk was a good membrane to extend the shelf life of food. And so this is how the use of silk as an edible coating became a reality. We kept investigating how this worked. And it's very simple. So basically what you're doing, you're starting from your silk, you make a powder out of it that is water soluble, you disperse it in water, and then you either spray or you deep coat your food. And by doing that, you're able to extend the shelf life. And why is very similar to other food coating. So what you're doing, you're decreasing oxygen diffusion. You're keeping the oxygen out. You're at the same time, you're keeping the water in, so you decrease the evaporation of water, and also you're decreasing the respiration rate of the cells inside the food. And the silk is also good to delay microbial growth. So it's not an antibiotic, absolutely, but it can delay microbial growth on the surface of food. And so this is just a slide that resumes what I just said. And when I published the paper, then I also learned about Appeal, which is a company that came out from University of California, Santa Barbara, that uses a different technology for food coating. And so I thought that could be a good opportunity also for us to become a company. And so I have to say Appeal was really instrumental for this technology, because they served as an example. And also when it came for our technology to grow, somehow they already went through the whole process of funding and showing the impact of their technology. So we were really happy to see that we could compete with them, and hopefully we can compete with them in the future. So I really just want to give credit to Appeal to doing an amazing work in this space. And so what we're doing basically is, what is really interesting is that the silk is known to be very safe. There are a lot of biomedical applications for silk. We do have FDA-approved silk applications, for example, for vocal folding, for suture thread. And so we work with the FDA to make sure that the silk was generally recognized as safe. And the same thing happened also in Canada, where it's considered a non-new food ingredient. And so now we can use silk as a food contact material, as a food ingredient in the United States. And so we basically try to understand how we could further develop this technology. So we try also to mix it with other polymers like PVA who are edible. And we saw that in this case, we can further control the barrier properties to the point that we can decrease the spoilage in fresh cut produce. And we started a company that is called Mori. Before it was called Cambridge Crops. And Mori is now having, the headquarters is in Boston, but we also have a branch in Salinas, California, where we're doing a lot of field works. And we have more than 50 employees. We raised more than 38 million in capital funding. So it's evaluated almost $100 million, if you like. And really, we plan to have a product on the market at the beginning of next year. And this is just to show you where the technology is at the moment, so how much we can enhance the preservation of food. It also works very well with meat and fish. And these are basically the product we're targeting now, where that goes from greeny leaves to fresh cut vegetables, cherries, fish, beef, but also candies. And really the push here is not only to extend the food shelf life, but also to minimize the amount of plastics that goes into the packaging. So I think at the end, I just want to thank my group, particularly the work that I presented today comes from some of my postdocs and some of my PhD students and all the funding agency. Thank you very much for your attention.
Thank you very much for that inspiring talk. I'm hoping that there might be some participants who want to ask questions, and I'm not really sure how this is going to work in this forum. Do they have to write their question, or can we hear them?
I ask all to be unmuted, and they can write their question into the chat. I think they can raise their hand and they can talk, whatever they want. I do have a question, so if nobody has a question, I can start.
Well, maybe we can just wait a moment. Are you able to see people raise their hands?
There was only one person, but it was only, I think Quinn was figuring out. He wanted.
So if anyone would like to start the questions, please raise your hand and we will get past this little technical slowness, since this is the first time we've done it in this way. Oh, I can see that there are people who are appearing, so that's wonderful.
Yeah, I'm muting.
You're opening.
Everybody one by one. I'm going to.
Okay. So at this point, people should be able to raise their hand and ask a question, and even also show their face to our speaker, which would be nice.
While we're waiting, I might go ahead and break the ice and ask a question. That was a fantastic talk, super inspiring. So thank you for that. I was thinking about the little needles that are delivering whatever you want to plants, and in my mind, I study pollinators, and of course there are applications of pesticides that are justified, but if those pesticides are systemic and they get into flowers, they're of course very bad for pollinators. So do you think your technology could deliver systemic pesticides to specific plant tissues? Like, could you get them into leaves and not into flowers? Is that a possible application?
So that's a good question. I think it would be possible. The problem is how you scale up to have an impact on, like it can work well in a lab, it may work well, relatively well in a urban farming settings, but then if you want to scale up, I think the application of this microneedle would be more difficult. And so on that side, spraying is working so well at the large scale. But in theory, it's possible. Like we're actually planning to do so. Like how can we specifically deliver pesticides or micronutrients to some tissues? But the application needs to be justified when it comes to cost and benefits.
Thank you.
I have a question.
Sorry.
Yeah. So what's the biggest barrier to implementation of your technology? They're all amazing technology. And I'm thinking, is that the problem of scaling up the technology, passing from the lab to the farm or to the market? Is obtaining raw material? It's policy change. Do you need to change policy and regulation to be able to use this material? or is people acceptance? Which one is the greatest barrier?
So I think you mentioned all the right barriers. I think so. There are several aspects. Scaling up of the material production is the biggest one. When I show you at the beginning how you go from the fiber to the solution to then the needle, it looks simple on a slide. It's relatively simple in my lab, where we can do kilos of material a week. But then if you need to meet the demand of agriculture, those are tons and tons of materials, and so that requires the right investment. And this is basically the latest series that the company had was to be sure we could have scale up. And then the second is how you source the material. Like silk is a commodity of China mostly, and so you need to be sure that the silk comes from China or from other sources. Like people are now trying to establish silk in Central America. You have silk production in Brazil. And so, for example, FDA is very interested in this silk that comes from Brazil for applications in humans. Because eventually, particularly if it goes in the food, we need to be sure that when the trees when the mulberry trees are cultured, they don't use too many pesticides. Otherwise, the pesticides will go into the silk. So those are the major limitations. From a policy perspective, I mean, obtaining the GRAS status was difficult, but not so much difficult based on my experience, because we already know that the silk is very safe.
Okay, thank you.
If I can just comment that I'd like to thank you for being the first CAFÉ speaker and giving a great talk to a general audience. And I was just wondering whether there were any other fiber compounds that this technique could be applied to.
That's a good question. It depends a lot on the you asked me basically if there are other fibers.
Yes.
So like, for example, keratin from wool.
Exactly.
So yeah, so I do have experience in using keratin from wool and doing similar material transformation, if you like. It's possible, but silk has some edge, particularly the regeneration from a fiber to the water solution is very straightforward and doesn't require harsh chemicals. And also in terms of properties, the fact that you can have a material that can be water soluble or water insoluble provides you with big opportunities. However, in my lab, we're now exploring new proteins, typically keratins, that you can use to achieve similar results, because it would be easier to scale up probably for some applications.
So there are some questions from the audience. I want to invite Janti Wuja first to ask his question.
Hi, I just had, I guess, kind of a more basic or fundamental question about the production that you guys use. So silk is typically associated as a pretty resource-intensive natural product, right? Silk textiles are typically associated with being pretty expensive. And so I was wondering how you grapple with that kind of cost benefit, and whether or not there are angles that you're looking at in terms of like maybe the synthetic production of silk, whether or not there are synthetic like analogs of silk that you can use, or what strategies that you're using in terms of production to kind of overcome that obstacle.
So that's a very good question. So we perceive silk as intensive and is expensive because of the textile requirements are particular. So they require cocoons that are very big, a single thread, and then the cocoons needs to go through processing for the coloring typically of the silk. In our case, we can use all the silk that doesn't meet the standard for textile companies and all the silk waste that the textile company have. So that really cuts down costs. I think the intensive part would be the shipment at the moment of silk between China and the United States, or Japan and the United States, or India and the United States. When it comes to synthetic silk, I think that's would be the future. Ideally, you want to have plants that grow your host, where you're using, of course, source of carbon that comes from wasted food, for example, or not But the problem is yield at the moment. So in California, you have Ball Thread, which is the company that is able to produce silk, spider silk in this case, synthetically in the largest volume possible, but still they have problems in meeting the volume required for agriculture. So until we're able to really boost production with synthetic biology, that will need to wait, because the caterpillar has been optimized through 5,000 years to make a lot of silk. The big opportunity for synthetic biology is that we can design our own silk. And so at the moment, we're borrowing a material that has been optimized for textile to use as a food membrane, but maybe we can design our own silk that optimizes the barrier properties. And so this is where we're looking for.
Thank you very much.
So the next question is from Jeffrey George.
Hi, great talk, thanks. So I was wondering about if there is any limitation in the protein or chemical or even the organism that you can integrate in silk, and as well about so you showed us about one aspect of the diffusion of some proteins into the silk, but I was wondering how fast it is like to diffuse across it.
So the limitations are, so we typically consider 50% as the maximum amount, 50 dry weight percent as the maximum amount of payload we can encapsulate in silk. And typically we operate with water soluble or water dispersible macromolecules. We can also do water insoluble, but it requires a little bit more of material fabrication. So it's more intensive, if you like. When it comes to the diffusion, it depends on the molecule you're looking for. Like gases are, and it also depends on the gas. Like if we have a silk that is very well beta sheet, the barrier properties for oxygen are very good. So why the silk works so well is that typically a polymer is either a good barrier for oxygen or is a good barrier for water, not both of them. Silk provides a good compromise between the two. And so in this case, it works well. But we need to look specifically at the molecule you would be interested to know whether or not it's a good barrier.
Thank you.
I'd like to expand on that question to ask one of my own, and that is when you are coating fruit, obviously the entrapment of ethylene could be really important not to have because it's going to promote softening. Have you considered formulating the silk so that you can have escape of ethylene,
so
but not entry of oxygen?
That's a very good question. And so I never been able to get the data for ethylene in my lab, so I cannot present them. But we do know that we can't allow the permeation, like the diffusion of ethylene. So we wouldn't have self-softening, if you like. So we don't have a detrimental effect on our fluid.
That's impressive.
But and this is all about, so basically every time we have this silk coating, we need to modulate the amount of beta sheet and random coils such that we can address exactly this type of problems. Do we prefer to have more ethylene going out or less water going out and such?
So what you're proposing potentially could reduce the need for refrigeration in many instances as well. I mean, obviously, if you have cooler temperature, spoilage is going to slow down, but in many parts of the world, that's not possible.
That would be a point. And another point is that by doing this, we could also allow shipment without refrigerated container, which sometimes are very expensive. And so you can think about opening new markets for some product.
So I don't know if there's more questions. I think I'll just ask one more since I don't see any. In terms of using rhizobium or other beneficial microbes in your seed coatings, have you evaluated the effect of this on colonization by the rhizobacterium and nodulation, for example, or just other changes in the microbiome, plus and minus the coating?
So I think we didn't investigate as deep as we wanted to. because I don't have the expertise. So we do see that the bacteria we're delivering, they actually do colonize the root of the seed. And we're doing now experiments where we're looking at the whole microbiome around the roots to understand what's going on. But I don't have an answer yet. But this is what we're actually doing this experiment now.
Yeah, I think that would be really nice to support your observations. Yeah, nice. Well, if there are no other questions, I just want to say that there's been, actually there is one, I'm sorry.
Yeah.
Robert Jinkerson has his hand up. So Robert, go ahead, you have the floor.
I have a question too later.
Great talk. I had one question about kind of on the market side. Do you see any pushback from vegans or vegetarians because you're adding an animal product to otherwise a vegetable? Are we going to have to see spinach labeled vegan spinach now because the alternative is going to have an animal product coating?
So that's a very good question. Other problems are, is this kosher? Is this halal? So it depends on how you would like to consider it. Sometimes the coating is so thin that in theory you could not disclose it, but in reality this is not what we want to do. We want to be very transparent, so we can go through the process of getting, for example, halal certification, kosher certification. When it comes to vegetarian, so basically to consumers that don't want to eat proteins that coming from animals, the alternative would be to use the synthetic silk in the long term. In the short term, you just need to be transparent. I have to say that there is an increasing amount of people who don't want to use silk because you kill the caterpillar in order to make the silk. But in our case, the caterpillar can come out, so we're not required to kill the caterpillar to obtain the raw material. So we can basically disclose all the process to the consumer, and then the consumer can decide what they would like to do. In other part of the world, I have to say, they don't care about this. They say, "Oh, I just want to be sure my food is eatable as long as we can." But you need to take care of all the different communities and the different cultures. So that, of course, is something we're doing.
Thanks.
Deb, go ahead.
Yeah, about Morocco, you mentioned that you had to do your field experiment in Morocco. Why is that? Why you cannot do it in US?
It was just convenient. Like we had a collaboration with them, and we exploited the collaboration.
Okay.
And now we're looking for collaborators in US.
Okay.
So to replicate the data and to get more data. So.
Okay.
Well, I am not seeing anything from others.
Yeah. No.
Yeah, but I will say that there's been a lot of enthusiasm and compliments to you for your presentation in the chat. And again, I mean, that was inspiring and just a perfect presentation that I think blended ideas and areas of research of many in our community. So thank you so much for taking this time to talk to us today. And I hope that an outcome will be people chat with you and potentially have applications that might result in collaborations as well.
Absolutely. Thank you very much for having me and for giving me this opportunity.
Thank you, Benedetto.
Thank you very much.
Thank you.
Bye-bye. Have a great rest of your day.